Hi everybody, it's Ms. Hamill with your 6th EOC review video. This focus is genetics. So you need to understand how organisms inherit their unique characteristics, describe how different forms of the gene are distributed to an offspring, explain how geneticists use the principles of probability to make Punnett squares. You need to understand the laws of independent assortment and the law of segregation, and how these laws apply to all organisms. You need to explain how the environment has a role on how genes determine traits, and you also have to describe how alleles from different genes can be inherited together. So here's some of the vocabulary, the key terms that you need to know. So a gene is going to be a segment of DNA that codes for a specific trait. So for example, the color of a seed. An allele is going to be a variation of that gene. So it would be the green allele, or the allele for the green trait, and the allele for the yellow trait. They are represented by letters, which would be the genotype, and we'll see that in a second. So the phenotype is going to be the physical description or the physical appearance of that trait. So a green seed would be an example. The genotype, as I said, are going to be the letters, the two alleles that you inherit for a gene. So they could either be capital G, capital G, capital G, lowercase g, or lowercase g, lowercase g. You always get two alleles for each gene. One comes from your mom, one comes from your dad. So one comes from the sperm, one comes from the egg. And when they fuse together, you have the zygote that has the chromosomes from mom and dad. You can pass these alleles on to your future children, and whether or not your kids express the allele depends on if they inherit the dominant or recessive gene from you. So a dominant allele is going to mask the recessive allele. It's always going to be stronger. The dominant allele is represented by the capital letter. The recessive allele is overpowered or masked by the dominant allele, and it is represented by the lowercase letter. Heterozygous or heterozygotes are going to be one dominant gene and one recessive gene. So big G, little g. This is called a carrier or a hybrid because they're a mix of both alleles. Homozygous dominant are represented by two capital letters. So two capital letters represent dominant alleles. They are called purebred, so big G, big G. And the homozygous recessive is going to be two lowercase letters. Two lowercase letters represent the lowercase or recessive alleles. They are also called a purebred. If you are a heterozygote, big G, little g, you are going to show the dominant trait because the recessive trait here is masked by the dominant allele. Okay, so Gregor Mendel and genetics, he is the father of genetics, so he came up with laws when he was researching heredity looking at peas. He had several rules. One rule was the law of dominance. This states that always the dominant allele will mask the recessive allele. So if you have a big G and a little g, whatever the dominant allele is, so let's say it's green and yellow, whatever the dominant allele is, it will show. The law of segregation, when gametes are made, the allele pairs are going to be separated amongst the eggs and sperm. So during meiosis, the gametes are going to form, and each gamete is going to receive the different genes. The law of independent assortment states that all genes are going to be inherited independently of each other, and this explains how alleles can skip generations. Also, this says that if you have curly hair, that does not mean that your hair is going to be blonde. The genes are independent of each other. So just to show you a visual of the laws, the law of segregation here, we have our red flower, which is represented here by the big R, so the red is dominant, and we have our white flower here represented by a little r. When we cross the flowers, we get big R, little r, big R, little r, big R, little r, big R, little r, and all of our offspring are going to have a probability or 100% chance of becoming a red flower, because big R, the dominant allele, will mask the little r, the white recessive allele. So they'll all be red. Then in the second generation, if we cross a big R, little r flower with a big R, little r flower, we would get one that is big R, two that are big R, little r, and one that is little r, and white. So what happens is that each flower has two alleles. The alleles separate in formation of egg or sperm. So let's say that this is one egg, this is a second egg, this is one sperm, this is a second sperm, and they are going to combine during fertilization, and the offspring will inherit the genes. The law of independent assortment here states that each gene is inherited independently. So it's going to be random which genes that are inherited for each individual. Okay, so in order for scientists to figure out the probability, they do what's called a Punnett square or a monohybrid cross. So as I said, these two letters represent a genotype of an organism, and each genotype has two alleles. So we have two parents that have big A, little a. They're both hybrids. So what you would do is put one of the letters above one box and the second letter above the second box. Then you would put one letter from the other parent on the vertical axis on one box and the little letter on the other box. Then all you would do is just match up the letters. So we have big A, we have big A, little a, we have little a, big A, but always read it big A, little a, because the A, big A masks the little one. And then finally we have little a, little a. So our phenotypic ratio, we have, if this is for normal and albino, the little a represents the albino. We have 1, 2, 3 normal genes, and we have one recessive gene that is going to be the albino trait. So the dominant offsprings do not have albinism, and the recessive do. The genotypic ratio is 1 homozygous dominant to 2 heterozygote to 1 recessive. So normal, carriers, and we have albino. So non-Mendelian genetics, so they don't exactly follow the law perfectly. We have codominance and incomplete dominance. So you need to understand the difference between the two. So codominance is when both alleles are dominant, so both are going to show. The example would be if we have a white chicken, a pure white chicken, and cross it with a pure black chicken, and it gives us black and white chicken. So the chicken like over here, which has black and white feathers. Both feathers are showing. Incomplete dominance is neither trait is completely dominant over the other, so they are going to blend together to produce a new phenotype. So the example here would be red flowers crossed with white flowers to give us pink flowers. And you can see that the incomplete dominants are represented with apostrophes. Another example would be if we had short and long phenotypes, and we would get medium. So it's an intermediate version or a blending of traits. Continued, we have multiple alleles, and this would be 2 or more alleles for one gene. I'm sorry, more than 2 alleles. All of them will have 2. So this is when we have more than 2 alleles for a gene. Blood type would be an example. So you could either be A B, or O. So if you're a type A, you are AA or AO. If you are type B, you are BB or BO. If you are AB, then you have AB blood, or if you are O, you have OO blood. Therefore, O is our recessive trait. The polygenic traits are going to be controlled by more than one gene on a chromosome. So for example, skin color, eye color, and hair color, there are multiple genes on the same chromosome that control the color, giving us a wide range of colors that we can have for our hair, eyes, and skin. Okay, so our sex chromosomes and our autosomes are going to be what makes up our genes. So we have 46 chromosomes total. 22 pairs are going to be autosomes, and the last pair are going to be our sex chromosomes. This helps to determine our sex. So if the last pair are XX, you are a girl. If the last pair is XY, you are a boy. Females can only donate the X's to their kids, and males can donate either or an X or a Y, and they are going to determine the gender of the baby. So if we have a mother who is XX and a father who is XY, you have a 50-50 chance of having a boy or a girl. So the sex-linked traits, we have traits that are carried, or a lot of times diseases that are carried on the X chromosome, and they are passed on the X chromosome from the mother. So boys have the trait more often than girls because the Y chromosome cannot carry dominant traits to mask the recessive one, because it's a short little chromosome. So girls are usually carriers if they have the disease trait present. Boys can never be carriers because they only get one allele that is either recessive or dominant. So if you look here, we have X, this is a recessive disease. We have X capital H, X capital H. We have a normal girl, X capital H, Y, normal boy, X little h, X capital H. We have a carrier girl, X little h, Y. We have a hemophiliac. So if mom is going to be a carrier and dad is normal, we have a 1 offspring or 25% chance of having an offspring with hemophilia. Last thing we have is a pedigree, and a pedigree is going to show traits of inheritance. So just a reminder that the circles represent a girl and the squares represent boys. So circles females, squares males, and the shaded boxes represent having the disorder or having the disease or having the trait. If they are partially shaded, that means they are a carrier. So this here is just a pedigree of the royal family. It's a sex-linked trait, and the males, you can see, are the only ones that actually have the disorder. So sex-linked traits are more often shown on males than females. Let's see if there's one more slide. Whoop. Chromosomal disorders. So there are other chromosomal disorders, and this is caused by non-disjunction, and this occurs during meiosis when the chromosomes didn't separate properly. So the gametes get too many or too few chromosomes. An example of this would be Down syndrome, which is trisomy 21. There are too many chromosomes on the 21st pair, so they have 3 on the 21st. And DNA mutations are 2 types, and we'll talk about them when we talk about mutations in DNA. We have frame shift. This is an insertion or deletion, and it shifts the entire frame, causes a major mutation. And then we have point mutation, which is 1 letter that is flipped. It may or may not have an effect on the phenotype. So that is our last thing for the genetics review. I hope this PowerPoint helped you out. Please study and take the practice quiz on Edline. Have a good evening.